In this talk, I want to show you how we use global quantitative mass spectrometry to do post-translational modification analysis. And if you have any questions about it, please interrupt me, and we can also make that more interactive. So the outline for this talk is that first I want to give you a short introduction, then continue with how we do PTM enrichment and what our current workflows are. Then I want to show you what depths of coverage we can achieve with our methods, in what databases that are publicly available all those global post-translational modification data is currently collected and available. Then how we do PTM site localization on peptides, how we analyze stoichiometry of proteins, of protein translation, post-translational modification, and later also how we do how we elucidate kinase-substrate relationships, and how we do network and pathway analysis. If you're interested in post-translational modifications on proteins, fundamental questions that might come up are which proteins are modified, which sites on those proteins are modified, and how do those sites change upon biological perturbation. All of the first three questions we are pretty good at with mass spectrometry-based methods. Other questions that are pretty hard to answer are, for example, what is the stoichiometry of these sites, and which enzymes modify these sites, and what is the mechanistic and the cellular function of those post-translational modification events. And with stoichiometry, it's really hard to do that with mass spectrometry or with any method, but there's possible ways to do that. And for the other three questions, you really have to design your experiment in a particular way to be able to answer these questions. Which enzymes, for example, you can do in vitro assays with kinases. For mechanistic function, you could study, for example, interaction proteomes after mutating certain sites. And for the cellular function, usually people correlate a cellular phenotype to a certain mutation that they do on the protein. At the BroadE, we have a strong focus on protein phosphorylation and ubiquitination. Protein phosphorylation is one of the most important post-translational modifications because there is more than 500 kinases encoded in the human genome, of which a large number are disease relevant, and there's more than 150 drugs currently in clinical trials against kinases, and therefore it's really interesting to do also unbiased global analysis of protein phosphorylation. Protein ubiquitination is catching up quickly with protein kinases, with protein phosphorylation, because there is an equivalent number of enzymes that modify ubiquitin sites in humans. And there's also now, since 2003, a drug on the market, which is called Velcade, that inhibits ubiquitin-mediated protein degradation, and it has been also approved. And as you see now on the next slides, the tools to analyze protein ubiquitination became a lot better in recent years. Therefore, it's getting also more and more interesting for us. If you want to analyze protein post-translational modification with mass spectrometry, the most typical sites you're looking at are phosphorylation, ubiquitination, and acetylation. To measure those modification events with mass spectrometry, you have to know that all of those modifications add a certain mass to peptides, and this mass delta we can measure actually. So, for example, phosphorylation adds adds 80 Daltons to peptides. Ubiquitination adds a deglycine tag that adds 114 extra Dalton to peptides, and this deglycine tag, as you see later, is generated after a tryptic digest. And acetylation gives us an extra mass of 42 Daltons. The amino acids on which those modifications occur usually are for phosphorylation, serine, threonine, and tyrosine. Also, aspartic acid and histidine get modified, but those modifications mainly occur in prokaryotes. And usually, aspartic acid and phosphohistidine are really difficult to measure because most of the methods we use for mass spectrometry are done at acidic pH. And those modifications are really labile to acidic conditions. So usually we don't see those. For ubiquitination and acetylation, those modifications happen on lysine residues. And if you look now at the frequency in a tryptic proteome, in a protein sample that was digested with trypsin, you only see that about 3% of all peptides are phosphorylated, and less than 0.1% are ubiquitinated and acetylated. And therefore, you have to enrich for those modified peptides. Popular enrichment methods are IMAC and titanium dioxide and antibodies for phosphorylation, a quite recently developed anti-deglycine tag antibody for ubiquitination, and an anti-acetylation antibody for acetylation. Enrichment methods for phosphorylation are shown on this slide. The most popular ones are IMAC, which is immobilized metal affinity chromatography, and titanium dioxide enrichment, and also specific antibodies against specific phosphomotifs like phosphotyrosine, and also there's certain phosphoserine antibodies that are also useful. If you look now at the pros and cons of those techniques, you see that for the metal-based affinity enrichment methods, we can get pretty good specificity. As for example, with IMAC, we see that in samples, 95% of all peptides after enrichment are phosphorylated, and only 5% are not phosphorylated. Therefore, we can achieve a really good coverage of more than 20,000 sites per experiment. And a nice thing about IMAC is also that the solvents we use during enrichment are really compatible also with other workflows because they only contain acetonitrile and trifluoroacetic acid and can therefore simply be evaporated. A disadvantage of IMAC is that peptide desalting is necessary before enrichment, otherwise the enrichment doesn't work properly. For titanium dioxide, the negatively charged phosphopeptides bind to the positively charged titanium dioxide surface. The advantage here is that this enrichment works in samples that contain urea, so you don't have to do desalting before enrichment. You can analyze more than 20,000 sites also with this technique. But a big disadvantage here is that you need high concentrations of glycolic acid or dihydroxybenzoic acid to prevent non-specific binding of non-phosphorylated peptides to the beads. And this causes a lot of trouble downstream with our liquid chromatography and also with our mass spectrometers. Therefore, the more popular way, or our main way to do phosphopeptide analysis currently at the BroadE is to do IMAC enrichments. But there's other labs that only do titanium dioxide, for example. Other methods are antibody-based. Really popular is also to use phosphotyrosine-specific antibodies that can enrich for tyrosine phosphorylated peptides. The pro here is that complementary phosphotyrosine sites can be enriched that you don't get with IMAC or titanium dioxide. But the big contra here is that you have really high input amount requirements. So usually for antibody-based enrichment techniques, you need more than 5 milligrams of total protein. And for IMAC or titanium dioxide, you can start with 1 milligram, for example. About 2 years ago, new antibodies have been released for the analysis of ubiquitinated peptides. The way that works is that ubiquitinated peptides, here you see the ubiquitin side chain on a lysine residue, get cleaved by trypsin, the proteins that are ubiquitinated. And as you can see here, the C-terminal end of ubiquitin is linked to lysine residue, and ubiquitin ends in a RGG sequence. So trypsin not only cleaves your protein of interest, but also the ubiquitin side chain. And the only thing that's left over after tryptic digest is this KGG residue. And you can use an antibody that is specific against this KGG peptide to enrich for those peptides. And this antibody is really nice. It works pretty well, and you can see that we can achieve enrichment specificity of over 70%. This enables us to analyze greater than 15,000 KGG sites in a single experiment. Namrata has even put this number to greater than 20,000 if you spend a little more measurement time here. And a disadvantage here is that you cannot distinguish mono from polyubiquitinated protein forms. And you cannot distinguish ISG15 or NAT8 from ubiquitin. But as ISG15 and NAT8 are really low abundant compared to ubiquitin, more than 95% of all sites that you identify are ubiquitin sites. We have to emphasize here that before this antibody was available, all of the workflows have been done on, for example, HA or flag-tagged ubiquitin versions, where you did the enrichment on protein level. And at that time, it was only possible to analyze a couple of hundred sites. And now with these new workflows, it's really a complete change that you can get this high coverage of more than 15 or 20,000 sites in a single experiment. Enrichment of lysine acetylated peptides works similarly. So there's an antibody that is directed against acetylation. This antibody doesn't work as good as the KGG antibody. So the enrichment specificity that we can get here is only about 20% or less than 20%. But still, it allows us to analyze up to 3,000 sites in a single biological experiment, which can be really interesting. And also here, we need large starting material, a lot of starting material. Between 5 and 10 milligrams are usually necessary. So how does a typical workflow look like for quantitative protein expression and post-translational modification analysis? So typically, our turnaround time is 2 months. And this starts usually with SALAC labeling, which can take up to 2 weeks. Then with sample preparation for whole proteome and PTM analysis. Typically, fractionation is involved because you have to reduce your complexity of your sample. And on the proteome level, we can reach up to 8,000 proteins. On the phosphor level, 20,000 sites, up to 15,000 ubiquitin sites, and 3,000 acetylation sites if you really go for deep coverage. The sample processing here usually takes another 2 weeks. Then we move on to mass spectrometry analysis on Q-exactive instruments, which can also take between 1 and 2 weeks, and continue with identification and quantification of peptides and proteins with either of the 2 software packages that Namrata has described before, which are SpectroMol and MaxQuant, and then do follow-up analysis and network analysis, which can take even longer in some instances. On this slide, you can see a typical workflow that we use to analyze protein expression and phosphorylation in a study where we're interested in 100 breast cancer samples. In order to be able to quantify those breast cancer samples one to each other, we need an internal reference that is present in each single analysis. So if you do, for example, an iTrack 4-plex strategy where we label each tumor by itself in one iTrack channel, we can analyze up to 3 tumors within one experiment. And to analyze 120, we have to do 40 iTrack experiments, in which in each of those we have then one internal reference that is always the same sample that allows us in the end to cross-compare and quantitate differences across all those tumors. How do we extract proteins from tissues? So usually we cryofracture the tissue, then extract proteins with urea buffer, do a lyc-trypsin digest, and this is followed then by iTrack labeling. After iTrack labeling, we can combine all of those peptides, separate them to reduce complexity, and usually we separate for phosphoproteome analysis the sample into 12 fractions, and for proteome analysis into 24 fractions for mass spectrometry analysis. Data gets then analyzed in SpectroMil and later on further analyzed in R packages, for example, to do clustering, classification, and differential analysis. For this analysis, we need about 1 milligram of total protein per tumor, which is about 50 milligram of wet weight tissue. What I want to show you is that all of the methods that we are using here are under constant development. And within the last year, we had significant improvements also in our coverage, both on the proteome and phosphoproteome level. For example, this SCX-IMAC is an experiment where we analyzed a luminal and a basal breast cancer sample and looked at the coverage for the proteome and phosphoproteome. SCX means strong cation exchange, so it's a different separation technique than this basic reverse phase separation technique. And this SCX experiment we have done in November 2011. And this basic reverse phase experiment maybe 7 or 8 months later with a different workflow. And we were able to improve our coverage for distinct proteins almost twofold, and also our coverage of phosphopeptides almost twofold within that time span. The reason why we have such better coverage currently is because Thermo has released new instrumentation, in particular UPLCs, so ultra-high pressure liquid chromatography systems that allow you to run columns with smaller bead size so you can get a better separation of your peptides. And then also those Q-exactive instruments were released that are just 2 times faster and more sensitive than the older instrument versions. And therefore, you get this better coverage. In addition, for the phosphoproteome, we also were able to improve our IMEC enrichment. The numbers that you currently see with 12,000 distinct proteins identified in tissues or 37,000 distinct phosphopeptides per replicate in a single experiment are currently the state of the art. So, yes, please.
So it appears that for multiple steps, there are variants of, like, for enrichment, you do different approaches. For digestion, you do different approaches. But what is the biggest source of variation? And how do those things affect coverage of phosphoproteins?
Right. So the question is, how do sample handling steps affect the variability in your analysis, especially in your quantitative analysis? And of course, if you do in parallel, for example, separation techniques where you fractionate your sample into your peptide sample into multiple fractions, or if you do in parallel your tryptic digest, it can happen that in one reaction, trypsin cleaves at a certain point and at a certain part of the protein, and in the other reaction, a missed cleavage occurs, for example, that you get a different peptide form, and this adds variability to your analysis. So the best strategy is to mix your samples together as early as possible with isotopic labeling, as Namrata has shown to you, with Salek quantification. If you can't do that, you always have to be aware, if you analyze your data, that those differences might occur. For example, problems with ITRIC labeling are that peptides can get oxidized on methionine residues, so you might have differential or oxidation of those peptides, and those peptides might be strongly regulated between biological states, and you just have to analyze then the oxidized version and the non-oxidized versions. And if for both versions the quantitative results cancel out, then it's an indicator that this is a artifact that was caused by sample handling and not a true biological effect. So it's always important to understand what's happening with your sample to be able to sort out artifacts that are derived from sample handling. But that's a good question, of course. Right. In this breast cancer analysis, we were able to also detect, for example, really low abundant transcription factors like the estrogen receptor, which was not possible 1.5 years ago, for example, in those discovery-based mass spectrometry experiments. I also want to show you that post-translational modifications can affect each other and can do crosstalk with each other. For example, for ubiquitination and phosphorylation, it's well known that ubiquitination can regulate degradation of kinases, can activate protein kinases, and phosphorylation can generate, for example, recognition signals for E3 ligases and also regulate the E3 ligase activity. For all of those reasons, it's usually interesting to get a holistic picture of the signaling events that happen in your biological model system. And therefore, we have developed new techniques that allow you to analyze protein phosphorylation, ubiquitination, and acetylation from the same biological sample in one workflow. And the way we do that now is after, in a Salek labeling experiment, for example, after we mixed the proteins, did a tryptic digest, separated out the peptides, we do serial enrichments. So basically, we enrich first for phosphopeptides, then for ubiquitinated peptides, and then later on for acetylated peptides. And the reason why that is possible is because only 0.4% of all post-translational modified peptides have different post-translational modifications on the same peptide. So it's only in really rare events you see peptides that have phosphate groups and lysine acetyl groups. Therefore, we really think it's a really good strategy to save sample amount because you can do those enrichment techniques from the same sample, and you don't have to do those experiments in parallel, how it's usually done in other papers. We have applied this workflow to in a study where we analyzed the effects of Velcade, which is a proteasome inhibitor, on Jurkat cells, which is a leukemia cell line. So those Jurkat cells were Salek labeled and in one channel treated with Velcade with 1 micromolar for 4 hours. And what you can see here in the scatter plot is the up and down regulated proteins. Upregulated proteins are here, downregulated proteins are here, and modification sites that are induced by Velcade treatment. So in this scatter plot, you have the different we performed 3 different replicates. You can see replicate 1 and 2, 1 and 3, and 2 and 3 plotted against each other. And as Namrata has shown you already on the proteome level, we only saw small changes. 57 proteins were regulated out of 7,000. But if you look at the KGG sites, at protein ubiquitination, you can see that strong effects are induced by proteasomal inhibition, as it is expected. And surprisingly, we see also effects on phosphorylation level that are reproducibly across 3 replicates. On lysine acetylation level, we don't see any differences in that study. What is important to note here is that it's really important to normalize the effect you have on modification sites like phosphorylation, ubiquitination, and acetylation for your protein level to be able to distinguish changes that are only due to upregulation on protein level. For example, if a protein gets upregulated here, also the phospho signal should go up. And therefore, we just normalize our effects we see on phosphorylation, for example, by the protein expression change. And what you see here are, for example, 10,000 sites that could be normalized this way and that are only changing on phosphorylation level and not on protein level. And that's an important point because in a lot of studies, if you look in the literature, full proteome analysis was not undertaken, and sometimes you really can't tell is it a change due to stoichiometry changes or due to protein expression changes. Right.
Let's go back to the previous slide. Can you just mention why these numbers are different than the numbers you showed previously? So these are replicate. These are the
right. So all of those are
triplicate.
right. All of those numbers are localized sites. So I will go into a localization of PTM sites later in a couple of slides and explain that. And all of those sites have been also normalized across protein expression. So not for all proteins, for all sites you have protein expression data. It's pretty good with a coverage of 7,000 to 8,000 proteins already, but there's still 20% missing. And the other reasons why they're a little lower is because it's overlapping fully localized sites across 3 replicates. So we only took for that analysis the highest quality part of our data that we could get to make solid statements about what changes are observed after Velcade treatment. In addition, in that study, we looked at the depths of proteome and PTM and post-translational modification coverage for different levels of input material and MS analysis time. As you can see here, for the high coverage, we used 7.5 milligrams of input material. And the reason why we used so much material was because we analyzed protein expression, phosphorylation, lysine ubiquitination, and acetylation from the same sample. And especially for ubiquitination and acetylation, you need so much input material to get those high numbers of modified sites. For protein expression analysis itself, only 300 micrograms of total protein would be necessary. And for phosphorylation analysis, we usually need something like 1 milligram of total protein. As you can see here, to reach 8,000 proteins per replicate, we needed 24 injections at 2.5 hours, which is 60 hours of measurement time. For the phosphoproteome, we took 30 hours of measurement time. And for ubiquitination and acetylation, we spent, for each of those, 15 hours of measurement time. We also wanted to find out how much measurement time or what coverage do we get with less measurement time. For example, for the proteome analysis, we dropped it down to 20 hours of measurement time, and still got 5,600 proteins. So this is interesting if you want to analyze more different biological samples with a higher throughput. So you could drop your coverage on the proteome level and instead measure, for example, 3 times as many samples, which can be interesting. And also for medium coverage with 4 fractions and 2 fractions on phospho and KGG, you still get pretty decent numbers, more than 10,000 sites on phosphorylation level, or more than almost 5,000 sites on KGG level. And what I was really excited about is that with a single injection of 2.5 hours, we can get now a phosphoproteome analysis of 5,500 phosphosites. 6 years ago, this was a Cell paper, actually, with that coverage. And about 3 years ago, it took a full day to get this level of coverage. And now we can do that within 2.5 hours. So we got almost tenfold faster within 2 or 3 years, which is a pretty good achievement right now in the field. if you look at the overlap between different replicates, biological replicates for protein and modification analysis, you see that the overlap is pretty good for proteins. It's about 94% among 3 biological replicates. The reason for that is that to identify the protein in different biological replicates, it's not necessary to exactly measure the same peptides, but you just have to measure some of the peptides of the proteins. The overlap among phosphorylation data sets is usually around 66%. The reason why it's lower is because you have to exactly identify and quantify the same peptides in all experiments. And it's even a little bit lower for KGG and KAC sites analysis. And the reason for that is probably due to a higher level of promiscuity between different lysine sites on the same proteins, because they often have similar functions. And usually we get something like 44% and 50% of overlap between for ubiquitination and acetylation analysis. All of those global post-translational modification data sets get currently curated in public databases. The best one out there is Phosphosite Plus. There's also others out there like PhosphoELM and PhosCEDA, but they don't contain so many data sets. The nice thing about phosphosite.org database from cell signaling technology is that it has information from over 14,000 studies, so it's really has a wide coverage. And what you see there nowadays is that there's more than almost 180,000 phosphosites reported already on 18,000 non-distinct non-redundant proteins. What we can definitely say already now is that more than 10,000 human proteins get phosphorylated in one form or the other. And if you look at the database itself, it reports sites that were identified via site-specific methods, so with site-directed mutation analysis or with site-specific antibodies in the past, and also sites that were only identified with mass spectrometry. And 93% of all of these sites have been currently identified with mass spectrometry. If you look at the overlap between both techniques, you see that there's only a 50 to 60% overlap. And the reason why we're missing out on some of the sites that have been previously characterized with targeted methods is because all of those studies have been done with trypsin to do shotgun proteomics. And only about 60% of all protein regions are accessible with trypsin. And therefore, you're missing out on some sites that are just not possible to get peptides for that you can easily analyze with mass spectrometry. On ubiquitin level, you can see there are 39,000 sites, and on acetylation level, 24,000 sites. Another problem with post-translational modification analysis is to localize the sites, as Carl has explained to you yesterday already. In this slide, I show you how MaxQuant takes care of this problem. So if you have, for example, 2 phosphorylation sites on one peptide, there's multiple different possibilities where those can be localized. And not for all of those peptides, we have enough sequence coverage with fragment ions to be able to tell where those sites are localized. And to be able to describe how good the quality of the localization is with a score, there have been different approaches. So in MaxQuant, they use a probability-based scoring algorithm where they just look at the total number of theoretical ions, which is n, then at the total number of the observed ions, which is q. All of them are fragment ions in the MS-MS spectra. And then also at the total number of matching ions. And what they calculate is a probability-based score that calculates the probability that a certain localization event can happen by chance. And usually they multiply it by -10 and take the logarithm to get a score. And the better the score, the better the localization is usually. In this example, there's 6 different possibilities how those phosphosites can be localized on one peptide. But if you have really long peptides with stretches of serine and threonine residues, the combinatorics can increase massively. And in MaxQuant, they go up to 100,000 different combinations to calculate those phosphosite localizations. So what they do is they calculate scores for all of those different possibilities, then calculate a fractional score. And the output that you get in the end is something like that, where after each residue, you have a probability score. And as you can see here, the phosphosite was localized there to this serine and this serine residue. And usually, if you have a score better than 0.75 in max quant, it's called localized in that software. It is also possible to analyze absolute stoichiometry for modification sites. So typically, as in the study by Jesper Olsen, you can see that cells that are in G1 phase, so most of the cells that you have in biological experiments, more than 50% of all phosphorylation sites have a stoichiometry, so a fractional occupancy of those sites of less than 15% or less. And only during mitosis, this occupancy increases massively because CDK1 and 2 phosphorylate all of those sites, and there you can see that more than 50% have occupancies of greater than 75%. To measure now the occupancies of modification sites on proteins, you not only have to measure and quantify the modified peptides where the site is sitting, but also its unmodified counterpart, and in the best situation also the abundance level of the whole protein. And you can do that with targeted methods, so with synthetic peptides that are isotopically labeled, and to directly quantify the relative amounts and calculate stoichiometries. But it's also possible now to do that with Starlag labeled samples, as it has been shown by Jesper Olsen. And in that approach, you use the different Starlag ratios to derive information about the relative occupancy in the different Starlag channels. Once you have your protein data, your post-translational modification data sets, and with regulated components, it's interesting what linear signature motifs are present in those data sets. Really popular tools that have been described in the past are from Steve Gigi's lab, which is called MotifX, which allows you to do linear signature motif analysis. We did that here, for example, for a data set where GSK3 beta was inhibited, and you see a proline-directed motif coming up. And other tools also allow, for example, the analysis of over and underrepresented motifs, like this ICE logo analysis. And what you can see here is not only the overrepresented forms of amino acids in a certain subset of your data set, but also the underrepresented forms. And this can be also really useful in some instances. Once you have your linear signature motifs in a certain subset of your data, it's interesting to link those linear signature motifs to kinases. There's about 520 kinases in a human genome that are encoded, and about 160 of them have one or the other linear signature motif. And you can use this information that has been collected in databases like NetForest, where classified phosphorylation sites were either derived from original publications or from in vitro specificity assays, where positional scanning libraries were used to identify linear signature motifs, and link that to your, overlay that to your data set. And what NetForest does, it also gives you a score of the goodness of your fit to a certain kinase. This works in some instances pretty well, but there's also problems here because some kinases also that are unrelated have overlapping linear signature motifs, and therefore it's nice to test your hypothesis also in a direct assay. For that purpose, we have developed a new assay where you can analyze in an in vitro kinase assay which proteins get directly phosphorylated by your kinase of interest. And we do that by Starlag labeling and cell lysis under native conditions. So all the protein complexes are still natively folded. Then we add the kinases and compare the abundance of phosphopeptides in the kinase versus the control state. And if we do that in a single shot analysis, we can quantify up to 3,000 phosphosites. And in a study that we did together with Nicolas Chevrier and Nir Heikohen, you can see that we have analyzed IRAC2, IRAC4, and TBK1. IRAC2 was not active in this assay, but IRAC4 and TBK1 were. So you can see all of those upregulated phosphosites due to kinase incubation. And if you look at TBK1, we saw here that among the 90 upregulated phosphopeptides, 45 of which had a serine-leucine motif. So we can say here that the TBK1 is really specific for leucine in the +1 position. And in addition to those motifs, you also get from that assay the identity of your proteins of interest because it's a full cellular lysate that you're analyzing. So it's a nice orthogonal assay to drug treatment data sets, for example, where you can just cross-reference the phosphopeptides and phosphosides to see what is up and down regulated under certain conditions. Once you have your linear signature motifs and also an idea which kinase might regulate it, it's interesting to overlay those data sets also with protein-protein interaction networks. There's a nice software tool which is called NetworKIN by Rune Linding's lab. And this allows you not only to match the motifs to your kinase of interest, but also to put it in the context of a string protein-protein interaction network. And gives you that information in the end as a score and allows you to generate new hypotheses. So Casper will go more into detail about protein-protein interaction analysis, as this is only a specific example for phosphoproteomics experiments. At the end, I want to show you online tools and references. So there's been a nice review paper by Josh Kuhn's lab, where the URLs of most of those tools are listed. So you can find them here. And a review I would recommend to you for reading if you're interested in signaling analysis by mass spectrometry and post-translational modification analysis is by Juna Kudari and Matthias Mann, because they really cover most of the methods that have been described here in this talk. Okay. So that's a really good question. The question is, in a setting where you, for example, have a sample where you want to get really deep coverage, the workflows we have shown here always use separation techniques. And the question is now, instead of, for example, separating out your sample into 4 fractions, measuring each fraction with a 2.5-hour gradient, why can't you just measure this one sample in a 10-hour gradient, use the same mass spec time, and get the same results, which would be better in the end because you don't have to do so much sample handling. But we've tested those methods, actually, and the advantage of doing separation techniques is that you can separate out really abundant peptides into single fractions, and those abundant peptides then don't interfere with analysis of low abundant peptides. And the separation techniques are so good that we have, for example, 90% uniqueness of peptides per fractions. So if you have a really abundant peptide that always interferes with a certain peptide in one sample, we can separate it out to 1 fraction, and then in the other fractions, other phosphopeptides are not affected by it anymore. But it's a really good question. So, but what we see is that fractionation is necessary to get the really deep coverage. Good. If there's no more questions, we continue with D.R. Mani, who is our statistician in the proteomics platform. And he will tell you more about how we analyze regulated components in data sets and do clustering analysis.